Okay, so what I thought I'd do tonight is a lot of students seem to be struggling with the information for the different questions to do with genetics. Let me just get myself set up. I thought I would do a little question based on that. So let's have a look at a question like this. So here I have, excuse me, a question which is telling us some information about varieties of cats. And what we can see with the variety of the cats that it's talking about is that it's saying that you have got the color for the fur, so the gene for the fur color is actually located on the X chromosome. So the fact that it's located on the X chromosome means that what we can see is we can actually talk about. I'd be right, I can't go through paper 2024. I'm afraid it's not able to be shown live. It's under lockdown. So we're not allowed to do it until OCR have actually released it to be available. So I can't do that, I'm afraid. It's not fair doing that as well, having those videos out there so that any students who are using those papers, for example, for mocks, they would be put at a disadvantage if I were to do it as well. So there's a reason why we lock down the papers and we don't offer them up for all students to be able to see it. So I'm sorry on that one. I'm happy to do other papers, but I can't do that, I'm afraid. But I'm going to answer this question. And so because it is telling us that the question, in the question that it's on the X chromosome, so we know that it's a sex-linked gene, and it's asking us to come up with some symbols, and it tells us that one of the alleles codes for black fur, and the other allele codes for ginger fur. And it's actually told us in this question that the two alleles are co-dominant. So it's a bit like your blood groups in the fact that your blood group is A and B or AB because AB is co-dominant. So we don't have one dominant over the other. It is multiple alleles. So in this case, we're told that the black fur and the ginger fur allele are co-dominant. And it's asked us to come up with appropriate symbols. It's asked us to construct a genetic diagram, and it's also asked us to distinguish between the fact that we have a female that's tortoiseshell, and the tortoiseshell has both colors, with a ginger male. So the first thing I would say is if it's black, I would do a capital B. So therefore, I would write an X with a capital B. And if it is ginger, I would have a capital G. And because I know that it is on the X chromosome, I draw it with an X capital G. So then what I would want to do is I would want to then bring that into, right, well, I've got a female. So the first thing I'm going to do is I know that I've got a female. So I know I've got two X's. And then I want to bring in what color they are. Now, I've been told that they are tortoiseshell. Therefore, they would have both of the alleles. Sophia, of course, let me just finish this question that I've got just now, and then I'll go to 23A on 2021 AS paper one. So let me just finish this question because I do feel like there's going to be something to do with sex linkage potentially, and this would be a way of them applying the knowledge. So what you have then is a ginger male. So let me just change colors. I want to do this one a different color. I've got a ginger male, so I know they've got the X chromosome with the ginger allele, and they're male, so therefore they have the Y chromosome as well. So what is the possible gametes that they would get? You'd have an X with a G, or you could have the Y, and with the female, it could be an X with a capital B, or an X with a G. So therefore, what you would do is you would then think about this with your Punnett diagram. If I do my diagram here, I can then bring in my X capital B, XG, my XG, and my Y. So then all you're going to do is you're going to bring them down and across. As simple as that. And so when you do that, you would obviously have X capital B, X capital G, X capital B, X capital G, and now I'm going to bring in the blues. I then bring in XG, XG. I've now got my Y here and my Y there. So therefore, oh, and be careful, that looked like I'd done an X there. I've got a Y. I have that these two are going to produce male cats, whereas these two are going to produce female. And then I need to look at, right, well, I've got a tortoise shell here. This one would be ginger. This one would be black. And this one would be ginger. So if they were to ask a question like that, that's what they're looking for. Hi, Paul. How are you? Right. So what was the question? It was 2021 paper. You're always causing trouble. Paper 2021. Let me just bring this down here. So 2021 paper 1 and it's AS. Let me have a look. Right, so we're looking for AS paper. Let's just go with that. Right, where are you? 2021, 2021, 21. Oh, and it was paper 1, wasn't it? So it's the red paper. Right, 2021, question 23. Let's have a look. Well, it's been really slow tonight. I think that's because I'm trying to do it in 2 different places. Come back. Give me a second whilst I get this up and running. 22, 23. Okay, here we go. So let's see. Sound is cutting out really. I think this is the problem that I've got in the fact that because I'm trying to do it with the TikTok studio as well, it's actually causing it to lag dramatically. I feel like it did that in my on the YouTube side of things. What is going on with that one? Right, here we go. Let's see if I can actually do this. It's really frustrating. Right, so oh, what is just going on? Oh, the web Zoom. I didn't want that one. What are you doing? Being so slow tonight. What's the best way to jump up a grade? Best way to jump up a grade is to identify where you are losing marks because is it that you don't know the content? Is it that you don't know how to apply the information to the content. What is it that you're struggling with the most? If you don't know the content, make sure you fill your gaps in the stuff you don't know. If it's a case of you aren't answering it in enough detail, then lots of practice. Use your mark scheme to find the keywords that's looking for, would be what I'd suggest. Paul, are you still in here? Seems better now. Perfect. Okay. So I'm trying to do so many different screens right now, trying to see if I can get it to help them. I got into trouble from TikTok the other day there because it was like, you're not engaging in TikTok enough. Not all right. Chill out. Right. Here we go. Sophia, if you are still in here, is this the question you are looking for? So this was the question which was asking you about, you've got a spirometer trace. So because you've got a spirometer trace, what is it that's going to actually happen with that spirometer trace? If I put you back in there, hopefully I'll do that. So then the question actually was asking you, if I can just get this question up again. So the first one says, explain why there is a downward trend. Unfortunately, the thing is, Paul, now people are stressing out because there's not enough time. So because there's not enough time, what they're doing is they're trying to find a quick fix. So they want the information. With most teachers, they don't have enough time to give everybody. They've already given them lots of time, hopefully. And therefore, they basically just want any quick solution. Right, so this first one then, it was asking you about that downward trend, and it's saying, well, why is it that you've got this downward trend? So it's looking for you to identify here that what you've got is, you know, that you're breathing, your tidal volume, you're going up and down, up and down. You know that that's the tidal volume. So you know with the tidal volume, what you've got is the breathing in and the breathing out. In, breathing out. Well, the fact that it's going downwards, that's therefore meaning, because what you've actually got is the volume of air is going to be changing inside the chamber that's being used for the spirometer. Oh, did you? Wow. So Paul had to go for this precise test yesterday. That's pretty cool. So what they tend to do is they put a nose clip, and correct me if I'm wrong, Paul, because you actually did this yesterday. So they put a nose peg on your nose. So therefore, you are only going to be breathing through your mouth. Now, this isn't an interesting one. I'm going to go on a bit of a tangent, but when I was giving birth to my second daughter, when I was giving it to my second daughter, they gave me gas and air to help with the contractions. Now, I had been doing hypnobirthing. And so when I was doing hypnobirthing, it was all about learning how to breathe properly in through the nose and out through the mouth. So it's and out. Well, when you get gas and air, what you have to do is you have to breathe in and out through your mouth and only your mouth. But I spent so much time teaching myself to breathe constantly through my nose and out through my mouth to be able to contain the contractions, that I therefore didn't actually take any of the gas and air in that was needed to help me with the pain. So because you can't be sure that people will always breathe in through their mouth, what they need to do is they put a peg over the nose so that you can only breathe in through your mouth. So by closing the nose, that ensures that the person is only going to be breathing in and out through their mouth. So what you do is you breathe in and out through the mouth, and this is connected either to a digital reader. So this is what Paul was saying on his one. It was connected to a data logger. So it was linked to the computer. There was a digital reading that gets sent to the GP. In the old style way of doing it, what you actually had was you had a chamber of water, and you then had this piece of equipment that was attached to that chamber of water, attached to the breathing tube, which was then linked to the person who they had a mouthpiece and they were breathing in and out. And what was happening is that was going up and down. And what you would get is there would be a different amount of air in the chamber. And so what you're looking at here with that downward trace that's happening, that's because what you've actually got is that volume of air in the chamber is changing. So volume of air in chamber changes. And that's because the carbon dioxide, so I'm breathing in and I'm going to be using the oxygen and I'm going to be producing carbon dioxide that's going to be breathed, that's going to come out. That's the whole point of ventilation. So we're bringing more air in, which has a high concentration of oxygen, which should diffuse into our bloodstream at the alveolus, and the carbon dioxide should be diffusing out, and then we're ventilating it. So the reason my rib cage needs to move up and out and then back down again is so that I'm changing the volume, changing the pressure, so I get air coming in and out so that I can maintain that concentration gradient. So we expect the carbon dioxide to come out, but the carbon dioxide should be being absorbed by the soda lime. So CO2 gets absorbed to the soda lime, and therefore that volume of air in the chamber will decrease, and that will give you that slight decline, that downward trend in the trace. And then if we carry on, oh, hold on. I've just missed all these questions. Sorry, guys, give me a second. Let me pop that out. And let's have a look at your questions. So Sophia, is that what the bit that you were struggling with? So the answer is about the volume changing because the carbon dioxide is being absorbed by the soda lime. What exam board is this? This is OCRA. Oh, hi, Chidima. How do you calculate heart rate? Well, heart rate is to do with the completely different organ of your heart, and you'd be looking at your pulse rate. So what you would do if you're getting your heart rate is you would either press down on your wrist just a little bit further down so you can actually feel the beat, and then you would count that within 15 seconds or count it within 30 seconds, and depending on whether it's 15 or whether it's 30, times it by 4, times it by 2. Some people do talk about doing it up at the carotid artery, but you've got to be careful if you're trying to feel your pulse up here because you don't want to cut it off for too long. So always go to your wrist first to work out your heart rate. We're actually talking about your breathing rate here. Could you also do, yeah, of course I can. Of course I can. Okay, so the next one was asking, one second, I have to move this across. So the next question says we're going to use this, but this time we're going to calculate the mean breathing, calculate the mean resting breathing rate. Right. So let me bring this back down. Move that down there. Okay, so now we need to calculate this breathing rate. And so what they're saying is this is at rest. So because they've told you in the question already that it's at rest, so the resting breathing rate, again, that goes back to your knowledge of you should know that your tidal volume is your normal breathing at rest. So therefore, because we've got our normal breathing at rest, we therefore know that we're only looking at this part here. So from here to here. So what we then need to look at, well, is how many beats do we actually get per minute? So what you then want to do is you want to say, okay, what have we got? This is all in 1 minute. So this is, and it tells you a spirometer trace at rest breathing for 1 minute. The trace shows a period of resting breathing followed by a maximum inhalation and exhalation. So therefore, what I then need to do is I need to count how many breaths did I take within that 1 minute. So always know that if you're counting it, if I was to start at this point here, I would need to go up and down and back to that one. 1, 2, 3, 4, try and find it out. 5, 6, 7, 8, and then if I'm going back to that point, would be 9. So in this case, if I'm looking at it, I can see 9, which is going to be breaths that I take, but you've got to be careful of your timing. So I know that this whole section here is going to be in 1 minute. So this is 60 seconds. So what am I looking at? I'm looking at how many breaths did I take in 60 seconds? Well, I'm actually only measuring to that point here. So then you have to look at your scale and work that one out. So we've got 10, 20, 30, 40, 43, 42.5 seconds. So in 42.5 seconds, I did 9 breaths. So because I did 9 breaths, but it wants me to get my breathing rate, and I would work my breathing rate into rate per minute. How many breaths did I do per minute? I would have to do a calculation on that one. So yeah, so if I do 9 divided by my 42.5, let me just do that again, 9 divided by, and then I times that by 60, that gives me my total breaths to be 12.7. Oh, hold on. And then did you ask for number 3 as well? Okay, so now the one thing I would say to you when you're doing these questions, to make sure whenever you're reading this, that you are using the mark scheme to guide you, and to also make sure that you are looking at the information that they've given you. When you print out or when you use a different screen, like I'm using, you might have certain scales that are slightly off. So be aware that when you're doing this and practicing it, you might get a slightly different answer to what is in the exam because you have printed it out at a different rate, because it has a different size to it. So this next one then, it's looking for us to talk about the using the trace, state the vital capacity, and they want you the answer, and this is where you've got to be very clever. They've asked for the answer in centimeters cubed, but they've given you the scale in decimeters cubed. So therefore, what they're looking for is, can you actually give a answer and do a conversion too? So they're wanting the vital capacity. So you should know that vital capacity is from the tip of the maximum inhalation down to the trough of the maximum exhalation. So therefore, we need to read across, and we know that each of these 5 is 0.5. So what I would then say is, I'm going to go opposite because it makes it a little bit easier for me. Come across here. So then I would say, and that's awful, Christine, what am I doing? Do not do that in your exam. Get a ruler. I'm trying to do this and do it a bit lazy, and I shouldn't do that. Use a ruler, because in the exam, if you don't use a ruler, you could, and that's what I did in the last one, you could get a wrong answer that doesn't match what they're looking for. So using a ruler, so here I go to the bottom. I will go up to the top again. Oh, crumbs. That's not what I meant to do. Why did you just do that? That wasn't very good. Try that again. There we go. Right, so now I'm going to remove my ruler. I've got my lines, and now I need to look at how many of these squares are going in between. So if I know that 5 of these is 0.5, I therefore know that each one is 0.1 decimeter cubed. So straight away, I've got that part of it that I need to use to help me to answer this question, and then I do the conversion. So it is about those different parts together. So if I'm calculating this then, 1, 2, 3, 4, 5, 6, 7, 8, 9. Oh my gosh, I've lost it. Try to do this on a small screen is really quite difficult. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 28, and I've got half on each side, so I would say 29. So then what I'm going to do is I'm going to times them together. The 29 times my 0.1 to give me my answer in decimeter cubed. And then what I want to do, obviously, is I then want to convert that. So, which equals obviously 2.9. And then I'm going to convert that into the centimeters cubed. So I'm going to now times that by 1,000. Oh, crumbs. To give me my 2,900. Now, I would always advise you, so some of you might be going, why did you need a calculator? And some of you might be going, why did you need to count it so slowly? I know when I skim read or when I look at a picture, sometimes the way I'm seeing it, it blurs slightly in my head. So I know for me that I have to take more care whenever I'm doing this. Yes, it is all recorded right now, and it is or should be on my YouTube channel as well. So therefore available straight away. So that would be how I would answer that question. Does that help, Sophia? You are very, very welcome if you're still in here. Um, oh, interesting. I'm going to actually just carry on with this paper just because you've asked me to do it and I haven't done this paper. Oh, that's not what I wanted to do. Go away. Let me do this next question because I think this is actually a really good one on practical skills. And this is why I say to those of you who are A-level students, not just AS, those of you who are in year 13, it's really important that you do look at the AS papers because the AS papers have got some really good questions that you can then use to try to test your knowledge on modules 1 to 4. Why do you keep freezing on me tonight? Right, let's try this. Okay, so if we look at this next question then. You're welcome, Sophia. That's a good question to actually ask on. In OCR, do we need to know about homogenization and cell centrifugation? No, you don't. That one is AQA specific. If they're going to ask a question on that, they will give you information about that in the stem of the question. However, when you did your TLC PAG, oh, I can't think off the top of my head. I want to say it's like PAG 7, no, PAG 7 is microbiology. Whatever one the TLC one is, when you did that one, you should have used a centrifuge and you should have then spun the sample, and what that will do is that will separate things out into their densities, and that means that you can get the supernatant or you could get the pellet at the bottom. So when you talk about homogenization, what you're trying to do is get an even spread of it all around. And when you're looking at cell centrifugation, you're just talking about trying to separate out the different fragments. But that's more AQA than it is OCR. Vital capacity is tidal volume and inspiratory. Yes, yes, it is. Right. So if we look at this question, then you are told in the question, let me just add in a little bit of information that they've given you. Oh, come on. That the ranges shown are the minimum and maximum value, and then they've asked you to describe the trend in the data. So they want you to describe the trend. Got you an oh, thank you. It's a funny one. Like trying to cover everything is really quite difficult on this platform because some students only use TikTok, some students use the YouTube channel. The YouTube channel saves it automatically. Although TikTok saves it automatically, if it is over an hour, which you know me, I talk forever, it takes me about another hour to download it to then be able to upload it, and I hate that. So that's why I moved over to do it the other way. And then obviously on Zoom means that people can join me and I will answer questions. But if we look at this question then, ask about the trend. So I like this because what you have to think about is the trend in the data shows that you can see a bit of a correlation that's happening. So if I focus solely on the boys, and I'm only going to look at the boys to begin with, and then I'll look at the girls next. I know Zoom is the best. That's what I use for my group tuitions and my courses and stuff. But I can live stream from the Zoom, which means I can put it straight onto YouTube. And also it means that if there are students that want to join and have these conversations, then I don't have to worry about those random people who jump in and try and throw you off course. However, you are distracting me, Paul, so I'm going to stop now. But if we're looking at the trend in the data, we can see that the trend is increasing slightly. So there is a slight increase with the numbers of, so we're looking at the maximum minimum range and we're looking at the resting tidal volume. So we can see that there is a slight increase from about the age 13, but as I've shown here, I've plateaued my line, and therefore that's showing you that there is a slight plateau. And so it's important that you actually state that whenever you're talking about what you see, and I ask you about trends. So with the boys, we can see that there is an increase, but then there's a plateau at the end. When we're looking at, for example, the ranges, it's important to note, look at how huge that range is. Look at how huge that range is. The boys' range is so much bigger than the girls, except when we're looking at 13 for the girls. The girls' range is much smaller, except to the age of 13. So these are the little bits of detail that you want to go into when you're given a graph like this. You have to really scrutinize the graphs that they give you, whatever they decide to give you, and it can be on any random bit related to the topics that you're covering. So it is important to be sure that you understand what that means. Okay, so what does it mean if standard deviations overlap? Now, these are not standard deviations. Just to be clear, this is not standard deviation. They've told me in this question that it is the range. However, what you've asked is a very good question. You said, well, what happens if you have your standard deviations and they overlap? So if I have, for example, a set of data, and I know, let's say my mean, whatever the data is, I don't know, 14.6. And my other mean is 19.3. I'm just making these up off the top of my head. So I have two means of two sets of data, okay? So that one mean, so let's say that's A and this is B. So the mean of A is 14.6 and the mean of B is 19.3. I've done all my calculations. I've worked it out. Well, if I have a standard deviation, and you should be able to calculate a standard deviation, if my standard deviation, which is plus or minus, is, let's say for A, 3.2, so that's the standard deviation of A. The standard deviation of B is plus or minus 6.3. So I'm just, again, throwing things out there for giving you a bit of information. What I'm saying is I am going to go up and I'm going to go down by plus or minus 3.2. So if I'm going up, I would have this up to 17.8. And if I was going down, I would take this down to 11.4. So that's my standard deviation for the variability in my data. Now this one is much bigger. So this one is going to go 6.3. So therefore I'm going to go all the way up. Obviously this is not to scale, by the way. I'm going to go up and I am adding in, so that's 6. I'm going to add in 6. So therefore that is 5, 25.5. And I'm going to come down to 13. Well, 13 is overlapping down here somewhere, actually, if I'd have done it right. So let me just read you that one. So if I go bring it down to 13, what that therefore means is there's an overlap all the way across here. So because there is an overlap, wherever those standard deviations overlap, that therefore means that there may not be a significant difference between these two means. But I can't be sure yet. Okay. I could say the fact that the error bars overlap would get me to assume that there is not a significant difference between the means of A against B. However, to be sure, what I would want to do is I would want to do a statistical test on that. So therefore, because I'm using two means, I would want to do a t-test, and it would be an unpaired t-test. And so therefore, I would use an unpaired t-test to be able to say with confidence that I have got a significant difference or no significant difference. So the larger your error bars are, and when we talk about that with standard deviation, the more variability there is within your data. So what you're saying is, how precise is my mean? Well, if I've got really large error bars, I cannot be sure that my mean is where it should be. My mean is obviously going to be calculated based on the data I've collected, but that doesn't definitely mean that that is the mean of the true value. And so that's where you've got to be very careful when you're looking at your error bars and you're looking at standard deviation. Always, always, always throw in there, I would like to do a t-test to be sure that there is a significant difference between the data that I have collected. Or if it is a correlation, so for example, if instead I wasn't looking at a two means, instead I was looking at, let's say, concentration and rate of reaction, and my data was following a trend that looked like that, then again, I would say I want to do a statistical test to see if there is an association between the concentration of substrate and the rate of the reaction, whatever that reaction may be. So it is important that you are looking at whether it's a t-test statistical test or whether you're looking at a correlation coefficient. So I hope that helps. Guys, thank you so much for those of you who are following me and who are liking and watching. If you have questions, please do ask. This is what I want to do, is I want to help you as much as I can whilst I have a bit of time. So I always go live on a Wednesday, and then it depends with regards to the other nights if I can go live. But if you have specific questions, please do ask away. Um, Yeah, I do have a stats booklet, Paul. Thank you. I do have, it's a module one booklet which covers the practical side of the assessment within the exam that they could potentially be asked. So yeah, there is a booklet that they can buy if you are interested. You can buy my workbook, which is on my website. I love it. Paul, you're like my marketing person. Come on, Christine, plug yourself. I always forget to. It's that whole thing of I want to help people, and then I forget that actually I still need to make a living. Do you think we should know about ecosystems examples? So, Musa, do I think you need to know about examples? Specific examples, no, because they've taken that out the specification this year, and they specifically said this year that they would not have students didn't have to know those specific examples, but to teachers still to use them as examples because it will be whatever they give you and you have to apply your knowledge to that. So it's definitely important that you know about the examples, but really what they want you to know, let me just bring it in. What the important part from any of the examples is, give me 1 second, let me just get up here's one I prepared earlier. feel like it's Blue Peter, not that you guys are old enough to understand Blue Peter. Are you, are you? So if, for example, I use the peat bogs, because I feel, I don't know why, I don't know why. I love that. Pepperoni pizza. Um, I feel like they could bring in the carbon cycle. And so if they're going to bring in the carbon cycle, they could link it to peat bog. So if they were going to give you an example, it could be looking at the fact that we have got in the UK these peat bogs. Now they have very rare and threatened habitats here because of the type of ground that's there. So the environment is really wet. You've got this spongy ground. If you've ever been there, it's really spongy and it's filled with decomposing vegetation, which is known as peat. So what we know is that that is a carbon sink. So this is where I think they could bring in the carbon cycle part of the ecosystems part of it. So the carbon sink is that the peat bogs will store carbon. We know that carbon gets cycled in the carbon cycle in the fact that the carbon dioxide gets fixed in photosynthesis. So during the Calvin cycle, that carbon dioxide is fixed to make GP. GP is converted into TP. TP is then converted into our any other substance, whether it's cellulose, whether it is starch, whether it's sucrose, whether it is going to be used as a respiratory substrate for respiration, which is then going to return carbon dioxide back into the atmosphere. So in this case, with the peat bogs, it's actually a way of removing carbon dioxide out of the atmosphere. So if we have peat bogs and we manage them and maintain them, we can actually store carbon, and carbon dioxide in the atmosphere could actually be a problem because that could cause our temperatures to rise, therefore causing our glacial ice caps to retreat, to melt, causing issues there. So there's a lot of interlinking going on here, and it really depends which way they want to go. But the fact that these peat bogs are these sinks of the carbon means that we are mitigating that climate change. However, what do we do when we dry it? We can then use it as a fuel source. So we then burn it. Well, what do? We release carbon dioxide back into the atmosphere. So because we're releasing carbon dioxide back into the atmosphere, that is therefore going to increase the greenhouse effect potentially and change the temperature. So there could be an overlink there. What does it mean for conditions within the environment? Well, it's really acidic. Remember, and this is where you have to link it to, if you have carbon dioxide and water together, what will happen is they will form carbonic acid. That carbonic acid can then dissociate into hydrogen ions and hydrogen carbonate ions. A lot of students forget the fact that the carbon dioxide and water reaction that you learned to do with the dissociation curve and the Bohr shift can come in any which place it wants to because it's talking about carbon dioxide and water interacting. So because of that, that's going to make it really acidic and also is going to make it anaerobic conditions. So if we've got anaerobic conditions, we're therefore going to have less oxygen available. There's going to be less organisms that can thrive if there's not a lot of oxygen available. So you're going to have some bacterial organisms, prokaryotic cells, which are going to be able to thrive in that anaerobic environment. So it's a really ecological niche. And remember, your niches are talking about the lifestyle and the habitats together for the organisms. So what do we need to know about biodiversity then? Biodiversity, supporting a wide range of those plants that need to be adapted to low nutrients, wet conditions, and also providing habitats and food for different animal species. So the animals that are going to be found there are going to have adapted to thrive in those areas. So that doesn't mean they're going to be the same animals that you would find in grasslands, for example. And then what can we do? Agricultural use, there's lots of things we can do. There's lots of sustainable management things that we can do as well. And if they're going to give you an example, they'll tell you little bits of information and then expect you to apply it. They do not expect you to memorize this information. That's not what they want you to do. They want you to be able to apply your knowledge to whatever they give you. So if I just randomly find a question, so hold on. That's what am I doing? I don't even know what I'm doing right now. Doing too many things at once. What am I trying to do? I'm trying to find a question on peat bogs that they could give you. So let me. Do, 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 do. Ria, I will answer your question after I have looked at this question. So I want to do, there we go. So this last question was in 2020, paper 3, question I'm going to show you. Nope. Try that again. There we go. This is the sort of thing that they will do when they give you a question like this. So what they're looking for is, can you actually use the information that they've given you to evaluate what a student has suggested? So what are they telling you in this question? Let's just have a look at this. Oh, come on, stop freezing on me. There we go. Right. A peat bog habitat has been damaged by peat extraction and by management of a neighbouring farmland. So we've got neighbouring farmland that's been managed. We've also damaged it by removing the peat because it's probably going to be burnt. Ecologists decided to treat the peat bog in the following way. So they've got a buffer region was created between the peat bog and the neighbouring farmland. There were no visitors allowed on the land. Ditches were blocked to raise the water levels. The peat was extraction, tree planting, and use of fertilizers were banned. A student suggested that this was an example of preservation. So then they're looking at, well, what's the difference between preservation and conservation? So preservation, remember, that one is all about there being limited interactions with humans in that area. So do you know the difference between when you preserve something to when you actually are going to use management techniques to conserve it? So I'm just add in a, here's one I prepared earlier for you again. So with this one, what is preservation? We're going to protect the species and we're going to make sure things are kept exactly as they are. How? Nothing is removed. No or limited human interference. For example, Antarctica, limited resources, limited people who are going there. So therefore, that is showing us that we are doing preservation. So if we go back to the question then, the fact that they actually stated in the question that there was no visitors who were allowed, that shows that it's preservation. And also the fact that they've said evaluate, you want to give stuff that supports, does not support. So the fact that we are going to have the no visitors allowed, that is a way in showing that it is preservation. The fact that you are banning the tree planting, the peat extraction, That again is an example of preservation. You're keeping things exactly are as they are. And then you've got on the other side, it not being preservation, the fact that it's conservation because we are managing what is happening by using management techniques, i.e., we're going to block those ditches to raise the water level back up. That is a management technique that is there for conservation. The fact that we're also going to be putting a buffer region between is management. So that's why what you're trying to do is answer the question that's in front of you. So you don't, Musa, to go back to your original question, you do not need to memorize the ecosystem examples. What you do need to do is you need to be able to apply the knowledge that you have over conservation, preservation, for example, like this one, to the situation that they've given you. So another thing they could do, now this one was another example. This one was actually in the practice paper. And to answer the question for those of you who've just joined the Zoom lesson, this isn't an actual lesson. This is just the free sessions that I offer to help students because the exams are so close now. So it's not actually a lesson. It's just me helping any student who has got their exam and they're studying OCR. Okay, so here is who am I? I'm Christine. So I am a teacher, a private tutor. I went to Cambridge University to do my teaching postgrad. I then became head of biology. I then became director of student progress. I then worked my way up. And I then had two very beautiful little girls, which meant that I wanted to change my career path. And so therefore, this is what I do now. I want to make sure that the skills that I learned through my 20 years of teaching aren't wasted. And so I do this as a way of helping for students because I know there's a lot of you out there who are really struggling with the A level and it's tough. Okay, just to be clear, it is tough. And it also is going to take time and effort. Um, the thing is, I am somebody who didn't find learning easy. I still don't find learning easy, to be honest. I struggle and I am very visual and I have to do it again and again and again. And so therefore, I know for some of you, the fact that I can offer the repetition means that they are hopefully going to learn something. And also, if you haven't done so already, do check out my YouTube videos because the whole course I put out there. I wanted somewhere to direct my students to when I first started teaching A level again, because I was teaching international baccalaureate at the last school I was at before moving over to OCRA. But I wanted to direct my students to good resources, but also I wanted a fast way of double checking what I had to teach, and I couldn't find anything out there. There are a lot of teachers who have just come into the profession who are having to retrain themselves on their A level that they took when they were at school. And so I wanted to offer something that was high quality, accessible, and would help anybody. And that's basically what I do and what this is. And so I do it on TikTok and I do it on YouTube. It's funny actually, because I actually in what are we on the 21st, so in 3 weeks time I will officially have been on YouTube for a year. That's crazy to think that it's only been not even a year yet of me doing what I'm doing now, but it's been so fun and exciting because I'm learning new skills. And also all the diagrams that I use and all the pictures that I use are my own. Ones that I just randomly take when I'm out wandering around and what I see and things like that. And I also learned how to use Illustrator so that I could then make diagrams that were easier for students to follow. So I hope that it is helpful. And yeah. Right, to answer a question like this, so explain why your improvements would help conservation of the peat bog. Oh, thank you. It's really important to know a lot of the time when they give you lots of information, and I hate that because I hate reading the stuff, but when they give you lots of information, the important thing is that you take the time to actually really dissect what they've said. So make sure you are reading the information that they've given you because they've given it to you as hints. That's the biggest bit of advice I could give any student is take the time to slowly work your way through the yeah, to work your way through the information because there are some hints in there as long as you take the time to look for them. So yeah, that would be my advice. Now, going back to the question that you have about enzyme practicals you need to know. I couldn't tell you the ones that you need to know. They could ask you any question whatsoever. So they can ask you about amylase, they could ask you about catalase, they could ask you about proteases, they could ask you about lipase. They could give you any practical assessment that they want, as long as they are linking it to the content that you know. It's not a case of that you have to know this one specifically, because each of you will do something completely different with your teachers because of resources that are available, because of what your teacher feels comfortable doing. So the content is the same, the practical isn't, unless you're doing other exam boards. Like if you were doing AQA, they have very specific required practicals. OCRA do not have that at all. Right, I am going to finish for the live on TikTok now, and I'm going to do that because I've almost hit an hour, and if I go over an hour, it will literally kick me out. Guys, thank you so much for following me on here, and I will be back again. Oh, actually, I've got my course on Wednesday and Thursday next week. So, and my group tuition on Tuesday. I might not be on until Wednesday evening next week. So I'll try if I can do as much as I can. um with regards to the free stuff that I can offer, but obviously I have to prioritize my living. My children need fed and all that. And so I will try and jump on here again, but I'll definitely come on Wednesday next week. And if I can, I will do a bit more. But if you're only just coming across me, please do go check out my YouTube channel. Every single playlist is linked specifically to your specification modules. And so therefore you can actually go and watch all my videos. The whole course is there. The only thing I would say is module 4 for the biodiversity conservation. I put in module 6. I need to move parts of it across, but I linked that together. But it is there, but I did it as a holistic way of because of how I teach. So do check out module 6 for that one. Thank you for joining me, guys, and have a good evening.